Hello, welcome to Thermo Fisher Scientific 4D STEM webinar series. This is Çiğdem Özsoy-Keskinbora, Product Marketing Manager at Thermo Fisher Scientific. We have Professor Chen with us today. He will talk about the ptychography reconstructions and center of mass analysis in general. Professor Chen is an associated professor at University of Chinese Academy of Science, where he received his PhD from the Institute of Physics. Before, he was an associated professor at Tsinghua University. He worked as a postdoctoral fellow at Monash University and Cornell University. And today, his research focuses on development of new electron microscopy techniques, especially at 4D STEM, and the replication on functional materials. Professor Chen, the stage is yours.
Thanks, Chidam, for the kind of introduction and also the invitation. I'm very happy to share our recent work in this 4D STEM seminar. So today I'm going to talk about reconstruction methods within 4D STEM, and I will focus mainly on these atomic resolution parts, especially the ptychography. You can see using ptychography we can have ultra high spatial resolution, and also we can do some three-dimensional like depth sectioning and also dopant quantification. So 4D STEM is not a very new technique, and it's becoming like more and more popular nowadays. The basic setup of 4D STEM is similar as conventional STEM imaging. The only difference is that we use pixel array two-dimensional detector instead of bulk like annular dark field detectors. So we can acquire a diffraction pattern at each scanning point. So here is an example. You can see intensity variations within the central disk and also the dark field region when you move the beam around in the sample. So if you want to have a high quality 4D-dimensional data set, you need a good detector. Usually it should have a high dynamic range, fast readout speed, also single electron sensitivity. When you get this 4D-dimensional data set, you can synthesize numerically to get different imaging modes. So this kind of detector can be used as a universal detector for multiple image modes. So with the development of new detectors and also new algorithms, we can get a structure and electromagnetic field of your sample from a large length scale at large length scales from millimeter to some angstrom scales. Using this 4D STEM. So when you use a smaller converse angle or the nano-sized beam and the also called the non-overlap condition, you can determine the string, crystal grain distribution, and also crystal orientation distribution from this nano beam 4D STEM data set. And you can also measure like electromagnetic fields from this 4D STEM. So when you focus a beam more and get atomic size probe, the diffracting disk and center disk overlapped, and you can get the atomic resolution images. And especially when you combine with some reconstruction algorithms like ptychography, you can improve the spatial resolution to like even like a lattice vibration limit, and you can get three-dimensional information from this reconstruction. So first, reconstruction method using this atomic resolution 4D STEM is the integrated differential phase contrast, or IDVC, or integrated central analysis, or I-central analysis. So there is a very simple image mode, and you can you can reconstruct the phase of transmitting function from the two components of central mass or DBC images using this simple integration method. And I want to emphasize that this phase this image we get in the phase convolve with the intensity of the incident probe. So the incident probe distortion of the incident probe will be within this inch center mass. This method actually is very dose efficient, and you can even use a convention like a centimeter detector to get this DPC image. So it's widely used for low dose imaging of beam sensitive materials or biological materials. So I will mainly focus on the electrotachography method, since electrotachography uses the pixel-related diffraction patterns at different scanning positions. So it's these methods fit well with 4D STEM setup. And first types of this tachography called Wegener distribution deconvolution, usually WDD, or if you use some simplified wording like a single sideband or SB. So these methods are basically non-iterative, and it uses a focused probe with small scan step size. So it's similar to like conventional STEM imaging. And the data processing is very simple and not computational intensive. So you can even do some lab processing during the data acquisition. So this here shows one example. So the basic principle for this WDD is that you do a Fourier transform corresponding to the scanning point, and then we can get this Fourier space spectrum. And so you can have this so-called double overlap region using some these regions, and you can do some phase retrieval to get the structure of your sample. So more details about the theory and also experimental conditions can be found in these two papers. We usually we focus more on the so-called iterative method, like ePi difference map for maximum likelihood, because these iterative methods are more flexible, and we can include more mathematical physical models to consider experimental and perfection during the reconstruction, such as like multi-slice varying to consider like 6 samples in 6 samples. So we can also use some larger probe design, like ultra focus probe conditions. We will I will focus more on this in the following talks. For tachography, at the beginning, people usually assume this so-called thin sample approximation or the phase object approximation. You can see that the accelerated function can be written as multiplication between incident probe and the transmit function. So the phase of this transmit function contains the projected potential of the structure information. Iteration. This method is optimization method. We can make the calculated accelerated function matched with this measured diffraction intensity. So we basically we use these loops to get a convergent probe and object function when the data error reduces to a satisfactory level. So at the earliest stage, the electrotachography and first being applied to measure the electrostatic potential around these nanoparticles, and the spatial resolution was like 1 nanometer with phase precision of only 0.3 radians. Later, people also try to use this tachography to do atomic resolution, even under a scanning electron microscope. Also, it's possible to do this depth sectioning using a single projection to get this depth position of 2 carbon nanotubes. So all these early works used conventional CCD cameras. The parts by function, quantum detecting efficiency, and also the radar speed of these cameras were not that good. So the quality of or the performance of this ptychography may be limited by this detector part. So when we started do the ptychography, use direct detection camera EM-PAD, and we choose this same two-dimensional sample MOS2, and you can see from the synthesized annual dark field image from its 40 data set, we have these 1 angstrom spatial resolution. And then if we use ptychography reconstruction and same data set, you can see the spatial resolution can be improved, especially when we use large scatter angles like dark field regions of the diffracting patterns to do this reconstruction. And we can even see this single sulfur vacancy defect, point defect. The spatial resolution improvement mainly comes from these high quality diffractive patterns. You can see high structural information from the high scatter angles here, and these high frequency information can be retracted from the ptychography reconstruction algorithm. And also the residual aberration induced probe distortion can also be corrected. So using ptychography, actually, we can use a large overfocus condition as I mentioned. And the main requirement for ptychography is that we need some overlap condition between neighbor scanning points. So if we increase the probe size, like by using defocus probe, we can use a larger scan surface data here to maintain to keep the same overlap ratio. So when we use large scan size, we can cover the same scan region using fewer diffracting patterns. So we can get large field view. Here is one example showing the field view of 120 by 120 nanometer square. And you can see atomic structure details from both these domain wall centers and domain wall in this traced bilayer MOS2 sample. And the spatial resolution of this reconstruction is still better than 0.7 angstrom. So for the phase contrast imaging methods, there is an interesting problem because of this multiple scattering of electrons. And so for 6 samples, the contrast and the resolution of these the phase contrast imaging method will be affected, and it's hard to get interpretable image from 6 samples. For example, here by simulation from ptychography and high resolution TM, you can see the sample is thicker than around 10 nanometers. It's hard to get an interpretable image. So we can consider or model this multiple scattering by using a multi-size theory, and following the same idea, we can have this so-called multi-size electroptychography. So instead of using a single projected potential, we can use multiple transmit functions from different sizes, and also reconstruct these incident probe function. So the compared to the conventional ptychography, here actually we need to reconstruct more unknown parameters about the sample structure. So for here, first we use this distorted perovskite oxide sample to demonstrate this multi-size ptychography method. We achieved better than 15 picometer spatial resolution, and we can resolve this 0.59 angstrom peristylene project distances, and also we can see the pure oxygen columns. And compared to the high image, the spatial resolution is improved more than 3 times. And we also improve the precision to measure atomic columns here. For heavy elements, we improve to sub-picometers, and even for light elements like oxygen, the precision of atomic column measurements is around 1 picometers. Compared to the conventional annular bright field, and the improvement is significant. So to get a robust multi-slice electroptychography, we consider 2 main factors. First is the propagator. You can see this face of the propagator is proportional to the propagation distance delta z, and also the lateral frequency of Kr. So if one get a significant propagation effect, we need a large propagation distance for lower lateral frequency. So when we try to do this the depth sectioning, and there is an ambiguity, and so we introduce this regularization of the object layers to suppress high z-dimensional frequency at lower lateral frequency Kr. So we can control this regularization by using different weighting parameters. The second factor that is important is that for the multi-slice tychography, we need to reconstruct more unknown parameters for the object. So we need so that the reconstruction algorithm is more sensitive to the experimental non-perfections, such as the partial coherence. So we use a mixed state probe to model this partial coherence, and you can see this significant improvement when we use multiple mixed stage for the probe. And of course, we also use multi-slice for the object. When we have these 3-dimensional structure information for the spatial 6 samples, it's natural to correct this so-called mistilt of the sample. So, and we know that if the beam is not exactly along the atomic column, the projected image like HADIF or ABF will give us many complicated features in the image, and so sometimes it's even hard to determine the atomic position, like the oxygen here. But for tychography, actually, we reconstruct the structure layer by layer. You can see in the movie showing the different the face of different slices from the top to the bottom surface of the sample. And after doing some simple post-alignment, we can get a good projected image. I will also demonstrate this capability using experimental data with more than 10 milliradians mistilt. So multi-slice tychography is also more dose efficient compared to other phase contrast imaging like IDPC or conventional tychography. We can have atomic resolution for these metal organic frameworks, even using a dose of 10 electron per axial square. So we know that for IDPC, you can only work at thinner samples, and multi-slice can work thicker, much thicker samples. And so if we can use a thicker sample, we can improve the dose efficiency more than 10 to 100 times compared to this IDPC if we give the same spatial resolution. So we can also detect single atomic dopant in crystals here in advanced simulation using artificially introduced single dopants in these crystalline samples. And so you can see the dopant at different depths here, 8 and 11. The distance between these two dopants, 3 nanometers. These agree very well with our structure model. We also say and also quantify these oxygen vacancies from a newly nucleate oxide superconductor. Here you can see the oxygen along this direction, this side, not this side. And also along the depth direction, you can see the much weaker contrast here in this this oxygen column. We using this phase intensity, we can quantify the concentration of the oxygen, right? Because because the phase from the multi-stage ptychography is linearly dependent on the sample thickness and also on the number of atoms along the projection direction. So we can quantify here, and in this sample, we know that the main oxygen vacancies within this so-called the middle layer or the apical inner apical oxygen layer here. So finally, I want to show our preliminary test results by using a much faster detector, arena detector. This is a dedicated 47 detector with 192 by 192 pixels, and the maximum with our speed is 120 kilo frame per second, and it also has a large dynamic range. This is the first installation in China, and you can see from using this fast acquisition, we can even scan like large 1,000 by 1,000 pixels. This is the same. This is very close to the conventional STEM image scanning points, and the distortion between these two, due to the sample drift, is very small. It's very close, like 0.4 degrees apart from this 90 degree for this strontium candidate sample. And the single frame, the quality of single frame diffraction is high, and also we can see this big Kikuchi bands used by a few diffraction patterns. So using these fast detectors, we can do a much larger scanning region for electrotachography. So compared to the earlier works by using the MPAD and the second generation MPAD, we can have a much larger scanning region here. This example showing the tachography reconstruction using 1,000 by 500 different patterns. You can see the oxygen column clearly here. We have this high quality reconstruction. So using these faster detectors, we can get much better statistics, and we can even do in situ experiments using 4D STEM and also tachography. So here comes to my summary. So I mainly covered this atomic scale 4D STEM, and we can do many different phase reconstruction techniques, and they have different advantages. So here I want to emphasize that this multi-stage electrotachography can work even for like thicker samples. So, and the phase of this reconstruction also is linearly dependent on the sample thickness, and it can be quantified. So you can even use this to do tier series like tachotomography. There are actually some proof of principle tachotomography works. I want to thank all my collaborators. For the early works I performed at David Monaco at Cornell University when I was a postdoc. Yi Jiang and Michael Austria also contributed a lot to the tachography algorithm. And when I came back to China, I also have worked with many of our colleagues to get different samples, and also I thank my students. So the institute And finally, I want to say a little few words about our institute. We have a good aberration corrected microscope with fast detector and video holders. Also, our institute is the open institution, and we have many international fellowships. And so, if you are interested, please contact me. Thank you for your attention.